Hey everyone, and long time no see, I guess. Here's a little treat for you for remaining subscribed to this channel, I guess. Here is an unproduced podcast I made for Coxgazart. I made it with myself, Sally LePage, and David Hewlett. This is a look at more in depth on some of the science and some of the background of one of these videos. I just want to say up front that Coxgazart had no control over the content or the editing or production. Unfortunately, the project isn't going to move any further than this, but who knows, if you really like this stuff, let me know, and maybe we'll consider doing some more episodes in a similar vein, I guess. I don't know. With that said, I hope you enjoy it. It was, I think it's a really good podcast. It just didn't quite fit the tone of Coxgazart, so I understand that it didn't work for them. But I thought you might enjoy it, so here it is. Hope you like it.
It's like if we learned nothing from Hollywood movies, you don't get angry and nuke it.
In immunology is just this philosophical question because, I mean, can you define self?
Hello and welcome to the pilot episode of the
Unnamed Podcast,
where we break out of the nutshell and give our experts the chance to go into more detail about some of your favourite videos. I'm Dr. James Gurney, a part-time writer and fact-checker for Coxgazart and a full-time microbiologist.
I'm Dr. Sally LePage, a science broadcaster and former biologist.
And I'm David Hewlett, and I pretend to be smart on television.
This episode, we're talking about the reason why cancer is so hard to beat, and we'll be joined very shortly by our cancer expert, Dr. Kimberly Luddy. So along with myself, Dr. Kim Luddy was brought in as an expert to really get in and fact check this video. David, what did you understand about the video and the topic?
It blew me away. Cancer City, tumor towns, complex, sneaky, relentless cancer, how it evolves and becomes stronger as we try to fight it. I think perhaps maybe the most exciting for me was just how crazy cool our rock star immune system is.
The immune system is doing one hell of a lot.
It's scary. It really is almost like an alien entity that's sort of overseeing everything going on in the body. And at the same time, wow, like just the way it's evolving and trying to counteract these cancers, and the cancers are evolving and mutating and becoming stronger as we kill off the weak ones, and we're inadvertently making these things more deadly by trying to get rid of them.
I think if it was a sci-fi movie, we would be on the side of the immune system trying to take on the alien threat. If it was a dystopian movie, we would be on the side of the cancer trying to resist against this totalitarian army government system, like this all-powerful force that's trying to crush us.
It's very Orwellian. Yeah, it's
really
very Orwellian kind of, we're all got to stick to your cubicle, don't speak out, for God's sake, don't dress weird because something will come along, ask for a passport.
I mean, yeah, it's a very powerful story, although in this case, we want the cancer to lose.
One of the suggestions I had for the earlier point of the script was instead of casting it as Tumor Town, you know, a bunch of preppy rebels, we should frame it as a billionaire coming in and trying to like get around all the local laws and restrictions and try to do their own thing. That didn't make the cut. The cancer here is clearly the bad guy, and we have these legal systems which in our bodies are the rules the system uses to kill tumors and other pathogens.
It was a great analogy. I mean, certainly for someone like me to perceive what was going on, because I love the way they added the complication of building permits. I thought that was a wonderful part of it, because it isn't just sort of like a battle between, you know, bang, bang. It was this sort of conniving kind of like, ooh, look over here, look over here. Yeah, it's just...
And the mundane bureaucracy, the way you take over a city is through having the right paperwork.
Yeah, yeah. There's like, oh, nothing to see here.
I think I just also finished reading some Kafka when we were writing this script. So it was like the Kafka-esque nature of how the immune system has to work and how it self-regulates and all the myriad of ways it can get sort of sidetracked down. It was a slight influence on some of my notes for the video, certainly.
I'm really hoping to go into more of the immune system videos as well now. I definitely found myself drawn. It did a really good job of drawing me into that side of things.
Well, Dr. Kimberly Luddy is an expert on cancer and the immune system in particular. She's looking at immunotherapies. James, what do we need to know about the topic before going into this discussion?
I think the biggest thing that I can certainly explain, hopefully, is the idea of how cells communicate. How do cells actually know what they're doing and who they're talking to? We'll probably talk about receptors that cells put out on their cell surface, okay? Part of the reason is obviously a cell doesn't have eyes, it doesn't have ears. How does it communicate? How does it know where it is? It puts these bits of protein outside which have a very specific shape. And that shape will fit some molecule. Let's imagine it's something like salt. So there's a little salt receptor outside the cell. A bit of salt comes in, it fits perfectly into that structure. That then changes the behavior of the cell. And there are a whole host of millions of different receptors the cells can have. It's a whole topic, but effectively that's how cells will understand what their environment is doing and how they should react.
And that's how chemicals can turn on and off functions in a cell as well, right? Is that a chemical will bind to something on the outside of the cell and the cell goes, "Oh, something's bound to this little stick over here, my little hand waving around. Therefore, I'll go and switch on the factory that does this job, or I'll turn off the factory or conveyor belt that does this job."
Exactly.
It's a bit like a Rube Goldberg type of a system where something drops here and it drops something else over there. And well, I'm going to hit the microphone every time I get excited, which is a lot.
We've talked a little bit about, again, the factories these things are turning on. It's probably genes, change in gene expression. But aside from this, obviously, information is coming into the cell. The cell also has to tell other cells around it what's going on inside the cell. So these receptors also take things from inside the cells and put them on the outside. There's some great cooked-up videos about how these systems work, how your immune system recognizes them. Philip even wrote a great book about it. If you want to check it out, it goes into great detail. But effectively, it takes a little bit of what's inside the cell and says, look what I have. Is this anything? To the immune system. The immune system looks and says, that's fine, carry on. If it finds something it doesn't like, it says, you should not be making this. It might be a virus, or as we'll talk about maybe a bit later, a cancer. The immune system then freaks out and decides, I'm going to kill you. And that's how your immune system effectively works in a very brief description. It looks at what the cells are doing and says, that's normal, that's not normal, that's a bad cell, that's a good cell. And in the video analogy, these are sort of the building permits that we're talking about. These are these receptors. These are how the cell or how the cancer or how the tumor tricks the immune system into doing something incorrect.
James, was this an existing analogy before the video, or was it one that you had to come up with?
I believe this was an analogy derived by Philip. I could be wrong. I hadn't heard of a tumor town quite like this in terms of the analogy system.
I'd never heard of Tumor Town before today.
It's certainly, I can see it being used by the people before, but I don't consider it a particularly famous example of how tumors and cancers work. And that belies the issue of people tend to think of tumors as a monolith. That's true of all cancer, right? When we talk of cancer, the old phrase was they had the cancer, when in reality there's lots and lots of different cancers.
Or looking for the cure for cancer, as if there is one cure for one cancer.
Right,
or
exactly.
one cure for all of the cancer, because it is all the same.
It's all the same. When in reality, if I had a type of cancer, let's say I have a type of blood cancer or something like that, and you have the exact same type of cancer, the mutations that have caused that cancer could be different. So the treatment we'd have to apply will likely be different. So there's no single cancer, but even in the same subtype of cancer, they can be very, very different.
Within the same individual, the cancers can be very different to each other and can behave differently, and even in a short space of time. So the idea that somehow we've got these drugs that work on not only multiple people, but multiple types of cancer, it's pretty remarkable.
And that we understand the system enough to even tweak the immune system, and that is extraordinary.
The way that these drugs work is by being incredibly powerful poison. So when we're playing the game of trying to cure or treat cancer, we're playing against evolution, and effectively we're going to lose. the tumor or the cancer is going to become resistant to those therapies. There are cases, obviously, where it doesn't, and the patient gets better, fantastic, everyone's happy. Cases where that doesn't happen and the patient needs to carry on therapy, we can't just keep pumping them full of poison. This has led lots of researchers to look for alternatives, and a very promising field of therapy that has been currently being explored is the idea of adaptive therapy and how we might use the immune system to better treat and control cancers. We're going to talk with an expert in this field, and this is why she was brought in to be an expert on this video. It very much addresses this idea of adaptive therapy and how we might better treat cancers. Dr. Kim Luddy is a research scientist based out of the Moffitt Cancer Centre. She received her PhD in immunology from Trinity College Dublin and has authored over several dozen papers related to cancer and immunology. Kim and I actually share a lot of research in principle. We both work on similar projects in terms of adaptive therapy. In my case, it's treating antibiotic-resistant bacteria, and in Kim, it's looking at cancer. Kim, it's an absolute pleasure to have you here. If you could maybe start by telling us, what are you most excited about currently in your field of research?
So my background is in cancer immunology. So we use the immune system to combat patients' cancers and to try and eliminate their tumors. We're currently focusing on using evolutionary dynamics to incorporate those with immunotherapy in cancer to kind of modernize the way that we treat our patients and to extend the quality of life for patients that are on cancer treatments like immunotherapy.
And has this always been an interest for you? Was it as early as you remember, or when did you decide this was something that you wanted to pursue?
Well, cancer in general, I was actually literally a marine biology major in college as undergrad. And I had one of those illnesses that no one could figure out what it was. And for a little bit, they thought it was cancer. And so I was sent to Moffitt. It was actually doing my internship at the Marine Research Institute studying mollusks. And I was sent over to Moffitt as a diagnostic patient. And if any of you have been through that process, it can be very slow and very terrifying, especially when you're in your early 20s. And I actually got the benign diagnosis. I didn't have cancer. And so I had that golden ticket, and I'm walking down the hallway of Moffitt. If you've ever been there, it's this interweaving hallways like, a lot of medical facilities are. And it felt like everyone else was kind of going the other way, right? They hadn't gotten the golden ticket. They hadn't gotten the benign diagnosis. And so, my smile quickly faded and it just got into my head. And so I finished my internship that summer at the Marine Institute, and I went down to my college advisor's office and I said, I think I need to do something different. Like, I couldn't get these people out of my head. And so I switched over to cancer, which was a very long and difficult process because my skill set of snorkeling with scallops didn't really apply to, an immunology lab. So it took quite a while. But it was that initial moment where I had that fear that I could be, that was going to be the end of all of these dreams that I had. I wasn't going to get to achieve these things that I had been working towards. And then that relief was just mine and not everyone else's. And I wanted to find a way to share that relief with other people. And this is the only way, because I'm a science nerd, it's the only way I know how to do that.
What a fantastic story. That's amazing. Absolutely amazing.
So are you working with patients in clinics then, Kim?
So we do have some clinical trials. My specific day-to-day, I'm in the lab, so I'm not in the clinic with the patients. But what I work on are things like tumor boards, where the patient's information is brought to us, and then we inform clinicians on some ways that they could maybe augment how they treat the patient, and then that's brought back to the patient.
What exactly is a tumor board?
So tumor boards have been around for a very long time, and it's when a clinician has a patient that doesn't really fit the norm. They'll bring the patient's information in front of a room of specialists with all different types of backgrounds, typically ones who decide on radiation, what types of chemotherapy, those kinds of specialties. And they'll all get together and they'll look at all of the patient's information and kind of help guide the clinician on how to advise the patient. At Moffitt, we're testing out a new type of tumor board where we include evolutionary biologists and mathematicians and immunologists, and we all discuss ways of using ecological and evolutionary principles to help steer and guide the treatment of the patients. So it's just a way of getting everyone in a room together and focusing on an individual.
How many people can you talk to? I mean, how many people can you put through that system? I mean, that seems like it takes a lot of time.
We do one patient a month, to be honest.
Wow.
And it's a trial of a tumor board, which is a very unique thing to do. And it's not interventional, so it's not directly affecting how patients are treated. It's just testing to see if we can get the system up and running and things like that.
But ignoring the fact that you're working with really sick people, it must be quite fun being able to collaborate with so many different disciplines. So working with mathematical biologists and potentially working with biophysicists and working with drug experts and working from an evolution and an ecology background. It's quite rare within science that you get to work so broadly across disciplines.
It is extremely rare. And we all enjoy it, I think, more than we should. The ecologist, Joel Brown, who's on our team, actually studied squirrels, right, for most of his career. And now he comes in and he takes ecological principles and he teaches them to us, and we learn how to adapt them and apply them to cancer treatment. So his stories are fascinating.
Yeah, maybe don't mention that to the patients. The squirrel part.
I need to know, how do you take squirrel biology and apply it to a cancer?
Ecology is much older than cancer research. It's been around a lot longer. A lot of the models, a lot of the systems, a lot of the foundational principles of evolutionary biology and ecology have all been around much longer. And in cancer biology, we're kind of recreating a system that was already there. But the way we look at it is that tumor cells, they used to be your cells, right? So they used to pay attention to the tissue signals. They played along. They did what they're supposed to do. At some point, and we haven't yet defined it, they become a species. And so we look at it as cancer as becoming a speciation event. And so now they have their own fitness. They can undergo evolution. They can be selected for. They can be selected against. And they live in an ecology, right? The tumor isn't just tumor cells. A tumor has blood vessels and collagen and immune cells. And so it's an ecosystem. Some tumor cells live right along the blood vessel, right? Some live further away. So it's similar to an ecology of, say, a riverbed, right? And so a lot of the principles as far as extinction and preservation of species, we just take those and inverse those. So instead of trying to keep the species alive like they do in conservation ecology, we are trying to make tumors go extinct.
I like to think of it, we can take all the lessons we've learned of making mistakes in the environment, all the ecological disasters we've created, and apply them in a useful way to treat a cancer, or in my case, antibiotic-resistant infection.
Well said. Exactly, yeah.
So we talked about Squirrel Guy coming in on this tumor board and lending his knowledge. What is your viewpoint when you're attacking these problems? What are the questions that you're always asking of a tumor and of a patient and of the situation?
So my role probably for the past 15 years in every meeting has been just to say the word immune system multiple times. And everyone's like, oh, geez, you know. And especially before immunotherapy, it was the cool kid in cancer. We used to not be. But even still, you just have to remind everyone that the immune system is there and that it is an evolutionary process. It has evolved to be fantastic, and it is dynamic. And so my role is to kind of say, okay, but we've got to protect this patient's immune system because that's going to be what cleans up whatever the drugs leave behind. Or what can we then use of the immune system to come at, say, cells that would be resistant to other types of drugs? And we kind of use the dynamics of it. Because in evolutionary biology or in conservation biology, one thing that's really dangerous to species extinction is if you have lots of different attacks, right? So if you have, say, flood and fire and pollution and humans coming at you from different directions, that actually is more dangerous to your species survival. And so in cancer, that's what we want to do. So if we come out with something with chemotherapy, my job is to say, okay, they're going to eventually fail on chemotherapy. Let's now use the immune system because it kills in different ways. And it kind of represents a different way of attacking this really dynamic tumor population. So my job is really just to yell T cells at everyone.
You're mentioning T cells, killer cells, immune cells. Can you give us a little reminder or crash course for perhaps some of us as to which ones are which? Introduce us to the different parts of the immune system.
Oh, okay. So the T cells are kind of the classic cells that people think about. They're very good at recognizing foreign materials. So every cell in your body has a way of displaying what is inside of that cell. And so say you have a virus living inside of, say, a skin cell, your skin cell will then display part of that virus on its surface. And the T cells have evolved to recognize these foreign particles. And so they are very good at going out and hunting and killing the target cells, right? They can find them. They're very good at saying this is a target cell, this is a self cell, and they can kind of tell the difference between the two. And they've evolved to be able to do this really well. But they rely on that specific protein to be expressed, that specific, say, viral particle to be expressed on the surface. And it must match perfectly to the T cell. And so they're kind of a different classification than what I'm referring to as natural killer cells, which also kill target cells. But the way they recognize what the target is a bit different. They recognize cells that are really stressed out. They don't rely on just that particular particle to be expressed on the surface. So a stressed cell will start to have all of these flags on their surface. And then NK cell says, okay, you're not well, you're not doing okay. Let's go ahead and take you out. And so they have very different ways of recognizing, and they can feed off of each other. So a viral infected cell maybe will evolve a way to not be recognized by the T cell, say it downregulates its display of this particular viral antigen. So then the NK cells will actually recognize cells that aren't doing that correctly as well. So the immune system has evolved to counteract some of these ways that, say, viruses and other pathogens can hide from it. So NK cells are really more of a stress function, and T cells are more kind of a, say, foreign pathogen function, just to kind of simplify it.
So you're talking about immunotherapy and using the immune system itself and improving and augmenting the immune system.
Right.
And thinking about evolution at the same time, but using our own bodies to fight cancer. How do you do that? Because I'm also aware that if your immune system goes into overdrive, you get autoimmune diseases. And you've talked about these natural killer cells which go around targeting stress cells. I'm like, I'm super stressed all the time. Is it killing my own cells when they're stressed? Because it must be quite hard for the immune system not to attack us. So how do you help the immune system attack cancer without inadvertently attacking your own cells?
And without stressing us out.
And without stressing us out even more.
So you're touching on two very, there's the kind of the specifics in the drugs that we use, and then you really went to the philosophy of immunology, which are two very interesting things that we could definitely talk about. The first one is to turn the immune system against a tumor. We have all these different tricks. Some of them have been proven to work really well. Some of them are still being investigated. But one that's really common in the clinic right now is called checkpoint inhibitors. Like you said, the immune system will attack self if it is turned in the wrong direction. And in order to regulate that, it has all sorts of checks and balances within the system itself. And one of those things are called checkpoints. So an immune cell will express a receptor that, once bound, will actually tell the immune cell, hey, it's time to relax. You know, you're getting a little carried away.
I'm safe.
Yeah. And tumor cells will steal these ideas all the time, right? And so tumor cells will then bind these receptors and say, hey, look, we're safe. Aren't we friends now? So we just go in and block that. So the tumor cells can no longer tell the T cell, hey, don't kill me anymore. We block that interaction. So the tumor cells can't take advantage of these checkpoints. And so it's checkpoint blockade. So anti-PD-L1, anti-CTLA-4, widely used in the clinic now. And they were kind of our breakthrough where people started to say, oh, immunotherapy might work. So we kind of removed those checkpoints.
What does PD-L1 actually do? What is it interacting with?
So PD-L1, programmed death ligand, is expressed on tumor cells and a few other cell types. And it binds with a receptor on T cells. And it binds with PD-1, programmed death 1 receptor. And what will happen is it actually makes the T cells not responsive. So they can't kill their target cell. And it keeps them from actually killing the tumor cell. And so a tumor cell that expresses PD-L1 will be protected from that T cell. But once we block it, so we take, there's antibodies and there's small molecules, so these are drugs that are developed in the clinic, and they go into the patient and they will actually block the PD-1 from binding with PD-L1. And that allows the T cell to then continue on its journey and kill its target cells.
So in this case, the cancer cell, the tumor cell, has a little flag up saying, don't kill me, which is what the T cell normally says. If it has that flag, it's safe. And what you're doing is going in and just blocking that interaction so the T cell can carry on killing tumor cells and being active.
Yes. Although I would say that PD-L1 is maybe not a flag as much as a sword, because these T cells not only will stop killing, they will eventually go off and die.
So this is the programmed death, the D and the PDL. So
yes.
Huge topic. Why would a T cell die? Why would our own cells, outside of cancer, have a sword to kill our own immune system?
So it's to keep it in check, right? So the immune system is going to crank up when you get say, an infection. And when it does that, it's going to go out and it's going to then alleviate the infection if all things go well. And so once that happens, then the immune system has to turn back off. It has to calm back down. It has to come back to what we call homeostasis. It has to chill out. And so in order to do that, it needs to get signals. And one of those signals can be PD-L1. And its normal cells will express it as well when they need to.
There's a certain level of brutality here, though, because again, you described it as turning down the immune system or regulating it. What's really happening is that the immune system is being killed. The cells are being killed in this process. So it's this, there's language of, it just turned down and got regulated. Like, no, for that cell, it died.
Rather than hiring new soldiers for an army and then making them redundant, just kill the extra soldiers at the end of a war. We don't need them anymore. We'll just hire new ones if we do need them again.
It is brutal. I mean, the whole system, that was what I couldn't get over from all of this, was just how absolutely ruthless these systems were.
What was the big philosophical part of immunotherapy thing?
So you mentioned basically self and non-self, which is in immunology is just this philosophical question. It makes all immunologists philosophers, right? Because I mean, can you define self, right? And so historically, it used to be that immunology was always based on the idea that the immune system can tell between foreign and self, right? It was the stranger hypothesis, for lack of a better phrase, right? And so if you have this foreign invader, the immune system can recognize it and can kill it. Therefore, the immune system must be able to tell what is self and what is non-self. And that drove immunology for a really long time. And then in like the 1990s, Polly Matzinger, who was a fantastically interesting scientist, if you don't know her, she came up with this theory while she was herding sheep, but she had all sorts of jobs outside of being an immunologist. She actually tried to put her dog on one of her papers and everyone got really mad. But she's great. But anyway, in the 1990s, Polly Matzinger said it's not just about being strange, you also have to be dangerous. Right? So the idea is it can be foreign, but if it isn't harming anything, the immune system isn't going to react. And so that explained things like commensal bacteria that we don't respond to, right? And you can have immune reactions to a sterile wound, right? So if you don't have a pathogen, you still get wound response, right? So it started to move away from this, it has to be stranger, now it needs to have some danger, right? So it has to be foreign and cause damage. Then we started to realize, okay, we do have all of these commensal bacterias and we have kind of autoimmune regulation and tolerance, meaning that the immune system won't react to certain things. And so the philosophy became there's not really a straight definition between what you are and what other things are, especially the nice example, like I said, is commensal bacteria. Is that bacteria you? Is it not? Right?
And particularly with cancer cells as well. Is it starts off with your genome and then mutation by mutation, it's slowly becoming not your genome. But at what point is it something different? And you mentioned that they're like similar species. I come from an ecology background, and I think we have dozens of definitions of what it is to be a species. They don't all agree. And it's like trying to categorize a rainbow into distinct colors. Like you arbitrarily choose at what point it becomes a different species. So at what point does a cancer cell become a cancer cell?
Well, so first that analogy, rainbow, I'll cite you, but I'm going to borrow that because that is exactly what it is. You can't really distinguish between them. And a speciation event is not defined in cancer. We do not actually have an answer to that question. If we did, I think we would understand a lot about preventing cancer, which is a huge field.
I guess that's what I'm finding so fascinating here, or hard to get my head around, is that, I mean, our cells are mutating anyways, in that we are obviously evolving as time progresses. And the cancer cells are just doing it too quickly, or are we just deciding that those cells are doing the wrong thing?
The way to really look at it, at least the way the evolutionary biologists kind of helped me understand it, is that an individual cell as a species is evolving every time it's being selected for, right? So it can evolve really quickly. It can multiply very quickly. It can change very quickly. Tumor cells, even though they are part of us, they've kind of left this multi-organism idea and they become their own individual species. So they are now a single cell organism living inside of your body. And at some point, like I said, we don't understand when that is or how to really define that. But at some point, they separate out from that. So their evolution is completely separate from us as a multicellular organism.
That's just so frustrating.
Yes. And the evolution of it is so frustrating because, any clinician will tell you have this patient responding and you want to keep them that way. And they just don't continue to respond. Yeah. The tumor cells really evolve. And that's why the immune system is really, really just cool, because even though it's part of us as a multicellular organism, it behaves in a lot of ways like a single cell population, and that it, not in the classic sense evolves, but it changes in the kind of the colloquial word of evolves over time based on what it sees. So you're originally born with a diverse amount of T cells that can recognize all these diverse targets. And over time, as you get infections and the populations of those particular T cells that recognize that infection will expand and contract and form memory, and you get these changing populations over time, you're actually changing the milieu of your environment, right, of your immune environment, so that that system is changing as well.
It's such an amazing system. It's how the immune system controls this and how it builds this is beautiful.
It really is. I've never, I was unaware of, I was aware we had an immune system, but I was unaware of just how specific and this beautiful sort of dance that it does to solve these tiny little issues that we would not even aware of in some cases.
So what is the difference between a cancer and a tumor? Because I use the two interchangeably.
Okay.
And I'm not sure I should be.
So tumors can be benign, right? So you can have a benign tumor where it's really just a growth of cells that aren't supposed to be there, right? It's when it becomes malignant that it becomes, the C word, this cancer that can actually become fatal and something that we need to get at right away.
This is a great point to segue into the video, because when I came on to fact check this video, I came in on the second or third draft of the script, and the whole way through they were saying cancer, 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 cancer. And I pointed out, until it's malignant, I think most oncologists and immunologists and cancer biologists would probably agree it's a tumor up until the point it starts to divest itself out of the initial site where it occurred. But I was kind of hand-waving, like, that's my understanding of what the gap between tumor to cancer is. I am not a cell biologist or immunologist. I'm a microbiologist. So I always want to make sure we reach out to people who are experts in the field. When we reached out to you, had you heard about Cuxhazzat before? Was it the first time you'd heard of them?
I hadn't. No, that was the first time, yeah.
How did you find the whole process of being asked to be an expert for a popular science video?
Well, I was thrilled. I really enjoy science outreach. So at Moffitt, we're right there with our patients. We're not in a separate facility far away. I eat lunch right next to the families, right? It's all grouped together. And so the idea to get to be a part of more science outreach was, I'm going to jump up and do that right away, because it's how we understand what's going on with ourselves, with our family members. It's how we deal with the trauma of cancer. It's how we go to understand what our clinicians are telling us and how to talk to our family members about it. And so anything that I can do to help with those outreach organizations is going to be huge for me. And then I went on and I saw the videos and I thought, wow, I've seen some YouTube videos, right? And I was like, oh no, and we'll see what this is. And then it's really well done. It's well organized. It's, all the videos are accurate. It's such a visual medium, biology, when you can see it, and so the way they animate it, it's just perfect.
A lot of my students are upset that my lectures are not just 2-hour-long Cuxhazzat videos. I think when they find out how I've been involved with them such a long time, it's like, this is boring. Like, where's the nice image?
Well, how do you take the content of essentially longer than a 2-hour lecture? I'm sure the amount of topic could span an entire lecture series. What was the process like, Kim, of trying to narrow it down into a 10-minute video?
Well, I've cheated a little bit. I work with mathematicians, and so they build mathematical models. And so for the past 10 years, I've learned how to really narrow down what is actually needed. And the mathematicians say, actually, you see, we don't need this. It doesn't actually affect the outcome. And they've kind of taught me how to kind of narrow the focus into what's important. And we use analogies, right? I mean, each of these videos has an analogy. And whenever you're explaining, biology to people, it's our go-to way of doing it, And so you can only use so many analogies before people are really confused. So you have to pick the specific topic that fits with that analogy.
That's a constant concern when writing these videos is, have we pushed the analogy part past the point where it's useful? I think it's, especially with a topic like cancer, how much complexity do we need to make the audience aware of when we're writing the script? Was there anything you thought or would have liked to have been included in the video that wasn't? Was there anything you want to tell our listener about the topic that didn't make it in?
So in the video, we talk about the different phases of the immune system recognizing cancer. And we talk about this elimination phase where our bodies are recognizing very small tumors before we even notice them and are eliminating them. I think that the elimination phase is something we brush by because we get straight to the disease phase. And even in research, it would have been nice to discuss, there's a lot of kind of controversy in the beginning about whether the immune system was actually even doing this, and kind of the science fights behind it were quite fun. I really like that part of it.
My gossip detectors are going off. What's the hot gossip behind these scientists who are fighting with each other about what's going on?
She said science fight.
So this was a while ago, but it was, science fights are well, it's really just science, really. That's all science is fighting, right? Until you get to the truth.
It's the perfect explanation, yeah.
Yeah. I mean,
Very polite fighting.
Sometimes. Sometimes. But yeah, so the notion that the immune system could ever recognize and kill a self cell, even if it's malignant, was widely debated. And the thought that we have these little tumors popping up and the immune system can then eradicate them and is doing it all the time is really hard to prove. And so you kind of have the people who understand the theory and assume that this is what's happening. And then you have the people who say, well, show me, right? Because that's what science is, show, don't tell. And so the early experiments were not great. And the point where a baby tumor is kind of born and becomes malignant, like we said, it's hard to know when that happens. It's really hard to study because you can't measure it, you can't see it, and you can't define it. It makes it very difficult to study. And so there was a lot of back and forth that said it was called immunosurveillance, right? The idea that we're surveilling all the time for tumors. Now we've understood that it is an event that is happening and it has been shown more rigorously with experiments. But for a while there, you'd get some people who would just absolutely know it would not happen.
One of the lines that came up in another Kurzgesagt video you worked on, Kim, Your Body Killed Cancer 5 minutes ago, was that, so we've got these oncogenes, so onco means cancer, and these are genes that turn on the rapid multiplication of cells when you're young because you want to get bigger faster. The way you get bigger as a multicellular individual is to make more cells. But that if you're an adult, they should be switched off. And the immune system can know that you're an adult, see that the oncogenes are switched on and say, something's up here. How does the immune system know that you're an adult?
I have no idea.
Great.
No, we really, right. So you're talking, now you really like to get philosophical. This is quite fun. So you're talking about information, right?
Yeah.
And a lot of the modern biology, it's mentioned that we were first for a while talking about stranger and then danger for the immune system to recognize. But now we're talking about an understanding that we don't really have, right? We simplify things so even that we can understand as scientists. And what you're talking about is information. Where is this information stored? We honestly don't have that yet.
I am so glad you don't know the answer to that, because when we were going over the video, that was literally one of the things, like, we mentioned that the oncogenes get knocked out by the immune system. Like, I know a little bit about the immunology. Like, how does that work? I'm just happy I haven't just missed a big part of immunology still.
I love that you were talking about how scientists are telling these stories to themselves in order to help them understand it. Like scientists telling themselves a story, oh, well, the T cells know when you're of a certain age, know that you're an adult. And so that's when they start to tell things. Because obviously we're, as science communicators, and in making this video, we're often thinking about, okay, what's the best analogy to use here to make it clear? But then there are some limits of that. And I hadn't really thought consciously about the fact that we do this to ourselves, even like as experts, even when we're not trying to simplify the information to communicate with someone else, we're trying to simplify it in our own brains. We are still telling ourselves stories.
Absolutely.
It's just something I just completely not thought of.
I mean, we have to, at least for me, maybe I'm only speaking for myself, but I can't get all of it in my head. I need to classify things. I mean, scientists, we love to classify things. And that's because we're humans and we need to understand it, and we're not superhuman. And so we are using analogies and classification systems that are extremely imperfect all of the time. And it's not just the mixed metaphors I use when I'm talking to my husband about immunology. It is also what I have in my own head.
We also, in making the video, chose a very linear timeline to discuss when events are happening. But the idea that a tumor cell begins, it spreads out and then gets knocked down back to a single cell, goes up and knocked down back to a single cell, back and down. We did that. I think we talk about that happening 3 or 4 times. Obviously, these things can be happening in parallel, multiple tumors. When you help discuss these patient samples, is this often a question that the patients or the people who are fighting cancer, are they asking, like, how and when did this happen? You've spoken before about it feels like it happened overnight. Is that a common occurrence with these patients?
It does feel, I think, to a lot of patients that it's overnight. And that is because when your tumor is in remission, you're not sensing it, you're not having any symptoms, it's not measurable, but it's still there. And once you can feel it, and once you can measure it, it's there. And so it's a yes, no. It's a binary choice for a patient and a clinician. And time is going by between those, right? So you go into remission and then you come out of remission. That to them feels like a temporal linear event. And so even clinicians, we can't get to stop talking about the linear progression of cancer. And it's because that's how we, that's how we
perceive
understand
time.
things. Yeah, that's how we perceive time. But it's like a lot of when you're trying to achieve a goal. So imagine the tumor is trying to achieve a goal. And it's not a straight increase where you're just slowly getting better at something, right? Until you've mastered it. It's not like the tumor is just slowly getting better at being a tumor. It is actually, trying new things and failing, and then having a little success, and then, the ecology around it or the immune system around it or something, the human around it changes, and then it fails again. And so it's kind of more of a hilly path up to what would be considered success.
Well, it's like all things with evolution as well. It's really hard to remember, because we talk in academic language, teleological language, we give the agency and we give purpose to these things. And we say, the cancer wants to attack the immune or wants to survive. And it's not even trying. It's a lot of trial and error and failing and succeeding. But it's not like this individual cell necessarily is like, I've learnt that these building inspectors are coming, so I'll learn to forge these permits to do the document. It's just the ones that happened to do that happened to not die. And so it's really hard to remember that we are giving these cells agency, but they don't actually really have it.
It was the biggest stumbling block for me understanding evolution and seeing how other people like myself who aren't sort of deep into this stuff, there's an assumption that these things are working towards something or have not a mind, but a purpose. And I struggled to lose that as I got more and more into this stuff.
Yeah, we have to, even amongst biologists, we have to remind everyone that that's, that the tumor isn't malicious. We can't anthropomorphize it. It doesn't have a purpose.
Well, that's no fun.
But so if we're going to, because we've got to get them to understand using evolutionary principles for treatment, right? So if you continue to say, if the clinician comes in and continues to have that mindset that the tumor is trying to survive, and then it really leans into maximum tolerated dose strategy, which is classic cancer treatments. Patient comes in, we give them the maximum amount of drug we can that they can tolerate, kill the tumor, keep the patient alive. And it's been the way that we have treated cancer since the beginning of chemotherapy. And in evolution, people, they understand that that's a terrible idea.
Yes.
Right? But when you anthropomorphize the tumor, it makes you want to maximum tolerated dose. You want to just come at it with this huge hammer and just hit it real hard until it's gone, right? But evolution is saying, whoa, wait a minute. What you're doing there when you give that kind of dosing strategy is you are selecting for resistance. The tumor isn't trying to survive. You are making it survive. And you are giving it this really, really heavy hit that destroys most of the population. But those survivors are going to be really strong and really fit. And so to then get the clinicians to understand that we want to use evolutionary principles to steer the evolution of the tumor into a treatable state, it involves an understanding and an everyday reminder that they aren't malicious.
It's like if we learned nothing from Hollywood movies, you don't get angry and nuke it. If you nuke it, it comes back bigger and stronger. It's,
It's that evolution.
That's
I know
it.
we can't curse here, but there's a great phrase that works with, evolution's a nasty little thing
It
when you're
really
trying to
is.
get rid of something. Yeah.
Oh,
Yeah.
If it wasn't angry before, it is now.
Yeah.
Well, after that amazing chat, Kim, I want to know what is next for you in this field. I mean, we've had so many unanswered questions just because no one in the field knows. So what are the questions that you really want to answer right now? Where are you going forward with your research?
I do theoretical and experimental immunology. So on the theoretical side, I'm definitely looking into information theory and the complex system and how the immune system stores this information and how we can then use that information to protect for autoimmunity, but then also against tumors. And my experimental work, direct clinical translational work, is looking at using the evolutionary principles in combination with immunotherapy. And so we are looking for what we call double binds. The great analogy, to use one, is the owl snake that they use in ecology a lot, right? So if you have this collection of mice living in a field and you introduce an owl, the mice will get picked off by the owls. However, some of those mice will learn to run into the rocks and under the bush, right? And so then the owl can't get them anymore. And so if you introduce a snake that then lives under that bush, you've actually increased the predation rate of that snake by having the owl there. And so what we say for that is that if the tumor cells are the mice living in the field, and your first treatment is the owl, so you say you use chemotherapy, radiation, the hormonal therapies, the things that patients are already going to be getting. We then want to figure out, what is the snake? What do we put under the rock so that when the mice run under the rock, we have something there and we facilitate the efficacy of that second drug. So it's kind of with the steering evolution. And we want to use the immune cells as the snake so that the patients can come in and get the therapy they were already going to get. And then once they are responding really well, hit them with the second drug, which is the immunotherapy. And so I'm really looking at the specifics of which part of the immune system is going to be best for doing this, which immune cell type is best for doing this, which immunotherapy mechanism is best for doing this, and which owl to introduce, right? So chemotherapy, radiation, and then when. So that's why we rely on the mathematical modelers to help us determine when's a good time to do this, how can we use this to then prevent the emergence of resistance, to keep patients from progressing, to keep metastasis from forming, and do all of these things in a patient while they're still at what we call their nadir, while they're still responding to that primary bet. And there's a lot of evolutionary dynamics we have to tease apart because they know in ecology, if you introduce the owl and the snake at the same time, you're going to get a third mechanism, right? These mice are going to run into your house where you have nothing, right? Where you do not want them. And so you have to introduce the owl and snake at the right times during the ecological trajectory of the tumor. And so we're trying to figure out when are those right times. And it's probably patient specific. So we're also trying to scale up how we can then personalize these mathematical models that use these two treatments in this way. And so I'm trying to do all of those things along the way and hoping that we get where we need to be.
In the bacterial field, we call this a catch-22 from the book, the catch-22, and the idea that you put the cells into an impossible situation where if you do A, you get caught out by B. If you do B, you get caught out by A. It's interesting that the cancer field has a different analogy to what we're using. It reminds me of, I tried to sneak this into a paper once I got taken out, where I put a quote from Jurassic Park of, Jeff Goldberg saying, life finds a way. If you push the system in a way, if you don't cause extinction, you're more than likely going to select for something that gets around whatever you're trying to do.
Yeah, the first thing is getting the ego out of the way.
Yeah.
Right? Drug development, and especially I was watching immunotherapy, become really popular and get really effective. And while it was happening, I was walking around and asking, well, we're probably going to get resistance. Look what chemotherapies, look what targeted therapies, like, how are we any different? And it doesn't really get in the mind of when you're doing drug discovery. You think, I'm going to invent the cure. I'm going to discover the cure. I'm going to get this. And you have to not just pull your ego out, but you have to also be able to say to a patient, this drug isn't going to cure you. So we're going to use it in a way that will either extend your life or will eventually get to a cure on all these kind of complex dosing strategies. And it's really hard, and you have to get past that first. Then we have to say, hey, cancer, stop reinventing the wheel. Pest management people understand how this is done. Bacteria people understand how this is done, right? Antibiotic resistance has paved the way for using evolution in the context of non-science population, right? I think you talk to people, they get it. I have to finish my antibiotics. Antibiotic resistance is a problem, right? You guys have paved the way. And so we're really just kind of following behind. And We have to learn your field and then apply that to our field, which is really hard, as you know, when you're a specialist, to learn about the others. And so it's ego, it's time, and it's also an understanding between pharmaceutical companies, clinicians, patients, and scientists.
And because Kurzgesagt is obviously represented by a duck, all of the little characters in the videos are little birds. One last question for you, Kim. If you were a bird, what sort of bird would you be and why?
Whenever someone asks what kind of animal you'd be, it's a bird. We call them snake birds here. It's the birds that swim in the water and they fly, so they can swim for long distances underwater. They have a snake-like neck, so you just kind of see their heads coming up. They're very common in Florida. They have their wings out all the time, so they can fly and they can swim and they can walk. And I feel like if I could do all three of those things, I would just enjoy all of the time, right?
You don't want to be an owl holding a snake. That's not your...
No. No. I prefer fishing, I think.
Is that because you like to be multidisciplinary, if we're to go into it? You like to use information from all of these different fields all smushed into one bird?
It probably has to do with I was supposed to be a marine biologist, and I was supposed to be gathering my snorkel gear and going out on the boats.
Weren't we all? Weren't we all?
And now I'm going into the lab, so I look at these birds swimming along and then flying in the air and think, geez, that's the life.
That was Dr. Kimberly Luddy, a research scientist from the Moffitt Cancer Centre. So what did everyone take away from that amazingly nuanced and in-depth chat? David, you're shaking your head. There was so much to take in.
It really was. I mean, my mind was blown by the video in the first place. And then to actually have Kim sort of delve into it and, discover the sort of the evolutions that are happening in the cancer cells, how obviously similar they are to our own cells, that and the immune system, which I'm just blown away by as well. it is just extraordinary to me. Just amazing.
Yeah, because I am not an immunobiologist. I am not a cell biologist.
Weirdly, I'm not either.
I studied whole animals and evolution and ecology and behaviors of whole animals. And I was amazed at how many of these same concepts and principles came up.
Yeah. This is why you said to me the other day, you didn't study genetics because they didn't have behaviors. And I was biting my tongue because I was like, they 100% do have behaviors. It's the same concepts all the way down through life. And it's beautiful.
It does feel like that.
This is one of the things that I love about evolution is it has this kind of fractal property of every time you zoom into a different scale, you still see the same thing happening. You have genes within a genome acts very similarly to cells within a multicellular individual, which is what we were talking about, cancer cells within a multicellular individual. But that is also analogous to multicellular individuals like a bee or a wasp or an ant within this larger colony society. Yeah. And as she was talking about it, I was thinking, because there are cancers essentially of bee colonies where these individual bees have these mutations that mean that they reproduce at the expense of all of the other bees in the hive.
There are cancer bees?
There are cancer bees, yeah. Cape honeybees.
See? Mind blown again.
James, what was your favourite takeaway?
The thing I found most interesting was probably how in-depth the therapy really is for these patients. Not just the drug level, but the personnel involved in running this adaptive therapy for patients, having tumor boards. When Kim first mentioned a tumor board, I thought she was talking about a resistance profile. Or they take some cells and they see, oh, it's sensitive to this drug or not. No, they're talking about, we have multiple experts. I imagine all PhDs or MDs coming together regularly to talk about a single patient's therapy. That is a level of...
For months.
For months. It's a level of care.
And they have a squirrel guy.
Right? I kept thinking back to the squirrel guy.
Yep.
Did you think of a guy like wearing a squirrel costume, like a superhero thing, like Squirrel Man comes in, or was that just me?
I had squirrels on shoulders and in pockets. That was my...
I had everyone else in the room is in like suits or lab coats. And then the traditional ecologist who's got muddy hiking boots is dressed all in khaki and green and has wild hair from running through a forest chasing after squirrels.
And he keeps having to run off and grab a squirrel during the discussion.
Yeah. And everyone else is talking about all these cells. He's like, but I remember when I was trying to catch these squirrels back in Bermuda and has all these weird field trip tales to talk about.
Squirrel!
Analogies that somehow break the case. You know, he's like, oh my God.
That's it.
It's the same as a squirrel.
Of course.
If there was a TV show, you know, House MD, Squirrel MD, that would be the case.
I kept thinking of House as well because she's talking about all of these weird, unusual cancers and they're a panel of diagnosticians. I'm like, so your House.
It is very House. Yeah, that's true. Yeah.
And Sally, what about you? What was your favorite or the thing you resonated with most in our interview here?
I think the thing that's sticking with me, because I do everything that I think about at the moment is how do we communicate science in various forms and the power of story. And because we humans have evolved to learn information through stories, we know that we retain information better when it is told to us in a narrative form. And so the idea that scientists are telling themselves stories, like the idea, oh, the immune system just knows how old your body is. We don't know how it does that, but we're telling ourselves this because that story allows us to then create further scientific questions that we can test or design experiments. But it really comes down to that making conscious the assumptions that we have. Like, we're assuming that the immune system is able to tell how old it is, and that is the story, but being aware that that is a story that you're telling yourselves. And now I think I'm just going to go away and spend the rest of the week thinking, what other areas of my life are there stories that I'm telling myself, but I'm not aware that it is a story that I am telling myself. I'm treating it as fact, but it is actually just a story.
It's a construct.
That's fascinating.
I think you nailed it there with, this is how science works. We are storytellers. Scientists are our entire medium. How we communicate our science typically to each other is by publications. The best papers are good stories. The best scientific work is something you can follow and say, I understand how they got from here to here and why that's important. Whenever I teach my students how to write, I tell them, I want you to write in a way that convinces the reader that what you've done is interesting and important. And how do you do that if it's not via storytelling methods?
But it's also why misinformation is so rife, because it's so much easier for misinformation to have a better story than science. It's interesting you say that all science is good story. A lot of science is really dull and boring. So where we have the replication crisis, people don't want to repeat it. It's like, why is this important? Well, someone else has done it, but we need to show that we can do it twice. Doesn't really make as good of a story as scientists aren't telling you this one secret that will cure cancer and they're hiding it from you is a much better story. And so that's why that one spreads.
We cut the cure for cancer bit out of this interview, obviously, just for consistency.
James, please think of the comments.
What happened to science is fighting? I liked this whole pugilist kind of approach to that science was just people yelling at each other. I had this wonderful image.
To James and me, that was not a revelation.
Oh, really? That's you're just used to that.
Well, I think it's because
it's combative.
Well, science is people. Science is a bunch of people coming up with things. And when people talk about, oh, yes, there were multiple hypotheses at the time that science was deciding with, and then science went with this. It's like, no, two people were sat in a pub after a conference arguing it out over a bag of peanuts as to who's got the better idea. That's science.
That's great.
Well, what a wonderful conversation this has been. Unfortunately, that's all the time we have for today. Next time, why don't you tell us what video we should be covering next, and we'll try and get the expert from those on and really dive into the topic again.
The podcast is presented by James Gurney, David Hewlett, and me, Sally LePage. And the producer is also me, Sally LePage. And our favourite listener is you. Bird fact of the week: flamingos are the only species of bird known to experience existential angst.